Okay, adherent cells. Adherent cells are the type of cells that will bind to the bottom of a petri dish or a plate. So for example, if I have a plate like this, adherent cells, even though there might be plenty of media here up to this point, these cells will just simply bind to the bottom of the plate and attach themselves to it. The only time you will see them not being bound to the bottom of the plate is if they're dead or they're going through the process of mitosis. When they go through the process of mitosis, they pop off, the divide, and then the two cells that result from mitosis will reattach to the bottom of the plate. Now, adherent cells, they come from epithelial tissue. They're epithelial cells. So we're going to talk a little bit about the structure of epithelia. So epithelia is a sheet of cells that line the walls of cavities and channels, or in the case of skin, serve as the outside covering of the body. Beneath the epithelial layer is the basement membrane, sometimes called the basal lamina. The basement membrane separates the epithelial layer from the underlying supporting connective tissue cells termed the stroma. So let's just read a little bit and then we'll come back to this picture. Epithelial cells, which make up the epithelial sheets, have a basal and apical side to them. The apical membrane is interacting with the lumen of the cavity. The basal membrane is on the opposite side from the apical membrane. So the typical structure of tissue is cavity lumen, epithelial cells, basement membrane, stromal cells. So let's go back to the picture we have above. And let me first just kind of draw out what this looks like. So the basement membrane is nothing but a mesh of proteins that has been secreted by the cells in that area. So you have this mesh of proteins called the basement membrane, right? So the epithelial cells are bound to this basement membrane. Now they're bound to it by proteins that are on their surface, on the surface of their cell membrane. Imagine that it's the same type of mechanism as receptor binding ligand, except the receptor's ligand is actually some protein in the basal lamina, not some freely diffusing protein like a hormone or something like that. Right? So the cavity is over here. So here's my cavity here. So the cavity could be anything from the inside of blood vessels to other forms like the airways of the lungs or the GI tract, for example. Those are three good examples of cavities. Now, on the other side of the basement membrane, so let me just erase this part, you have the stromal cells. And we're going to draw them differently because they're actually a different cell type. So here are the stromal cells. And these are the supporting cells that are supporting the tissue and the epithelial cells. Okay, so these cells are actually a different differentiation status. They're different types of cells. The epithelial cells are different than the stromal cells. Of course, genotypically they're equivalent because they still have the same chromosomes. Now what we have here is a picture of basement membrane. So what is this? Epithelial cells coating the villi of the mouse small intestine. So it's kind of a little bit hard to see in this picture. First of all, the blue right here, so you can sort of see these little blue things right here and here, for example, and here, right? It's kind of like a bluish, purplish color. Those are the nuclei of the cells, okay? So those are the nuclei. And the basement membrane is stained red. So you can kind of see the basement membrane. Actually, you can see it very well. It's right here. See, this is the basement membrane. I'm just going to color over it with white. Right. So, and this, of course, goes all the way through here and right here. And right here. Right? So the stromal cells are here. These are where the stromal cells are, and this is where all the nuclei are from the stromal cells. And the epithelial cells are all the green. So everything here, like all this stuff, is the epithelial cells. A lot of epithelial cells, okay? And now you can kind of see the structure of the tissue, right? Because you have the epithelial cells facing the cavity, because the cavity is right here. So one side of it is facing the cavity, the other side is bound to the basal lamina, or the basement membrane. And that's the structure of tissue. That's the structure of tissue all the way across. Now, so the basal side of an epithelial cell is the side that is bound to the basement membrane. So B for B. So this would be the basal side. I'm just kind of showing it in blue right here. So this is the basal membrane. And opposite the basal membrane is the apical membrane. So the apical membrane would be in red right here. So apical. Okay? Just remember B for basement membrane, and apical will be the opposite of the basal side. Okay. And this picture right here is showing this even more clearly. Okay, so here you can see the basement membrane right here. And this is a collecting tubule of a kidney. And here is the basement membrane right here. Of course, this is the lumen, the cavity, right? And here are my epithelial cells, right? Here's one right here. Here's one right here. Here's one right here. Here's another one. And then on we go, right? The red right here, these are the nuclei, in case you were wondering. Okay? And the stromal cells right here, you can see them. They just look different, right? The morphology of them is different. So you can see that these are cells with a different phenotype. They're supporting cells for that tissue. In culture, in vitro, epithelial cells need to bind to a surface to survive and grow. Failure to bind to a surface will eventually trigger cell death. So the reason for this is actually kind of multifold. But one of the biggest reasons is this. So here I have the basement membrane, and here I have an epithelial cell. I'm going to draw it a little bit bigger, just a single epithelial cell. And it has some proteins that are binding it to this basement membrane, right? Upon binding, remember I said that you can think of these proteins that are binding the cell to the basement membrane as receptors, where their ligand is actually some protein in the basement membrane. Well, whenever you have a receptor bind its ligand, something happens to the cell. Some sort of signal goes into the cell, because otherwise it's kind of useless, right? What will happen is upon binding its substrate, its ligand, these receptors, which is the substrate being, or the ligand being some proteins in the basement membrane, a signal goes into the cell to tell it to survive. And these are called trophic signals. Okay, trophic signals, survival signals. So what do trophic signals do? Trophic signals basically inhibit apoptosis. So trophic signals will inhibit, let me scroll down a little bit, will inhibit apoptosis, programmed cell death. In the absence of these trophic signals, what will happen is the cell will actually get closer or be pushed through apoptosis. So it's very important for these cells to bind to some sort of basement membrane type of thing, or bind to something. Otherwise, they're going to die. The other thing is that among these signals that the cell receives, some of them are actually kind of growth promoting signals. Now, these are not going to be signals that actually by themselves will push the cell through mitosis, but in their absence, even if the cell is receiving growth signals otherwise from other sources, it will not be able to be pushed through the cell cycle and go through mitosis. So there's different types of things that are happening there, and that's why it's so important for these cells to have something to bind to. So it's important when we have this petri dish that we have this bottom part of the plate right here, that we create a surface that these cells can bind to, because if you don't create a surface that these cells can bind to, then the cells will be floating around in the liquid media and they will eventually die. So creating a surface for cells to bind to in vitro, a dish or culturing flask without a treating surface, relatively hydrophobic. So that's option one, okay? So you have your dish. It has a surface that's rather hydrophobic on the bottom. And what you do is when you put your media, which contains serum is a mixture of a lot of proteins. What will happen is the serum, the proteins that are in the media and that come with the serum, those proteins will coat the bottom of the plate. And that will create a surface for some cells to bind to. Some cells, that's good enough. They will bind to the bottom of the plate that's been sort of coated with some of these proteins from the serum. Now, the second option is a dish or culturing flask with a hydrophilic surface. And this is sort of like a progression. You first try the option A, then you go to option B, because each option actually gets more expensive. Okay, so you want to try option A with your cells. If option A doesn't work, the cells are not happy, they're not binding to the bottom of the plate, they're dying, go to option B. So in this case, the bottom of the plate is hydrophilic, so a couple of mechanisms can cause attachment. The serum in the media will coat the bottom of the plate, although the coating will be different than A, because what will happen is that you have a hydrophilic surface, so you could think that perhaps different types of proteins are binding to the bottom of the plate than a hydrophobic surface. The hydrophilic surface might itself play a role in attachment, meaning that for some reason or other, certain cells are happier with a hydrophilic surface and they bind more easily to it. So there could be a few things at play here. The third option is a dish or culturing flask with a treated surface. A surface coated with proteins found in the basement membrane, such as collagen. This treatment can be done by the experimenter, or treated plates and flasks can be purchased from a vendor. So in this case, what you do is you either buy some of the ingredients or proteins that are in the basement membrane or found in the basement membrane. You coat the bottom of the plate with it, and then once your plates have been coated, then you put your cells, and now your cells actually have something that looks like the basement membrane for them to bind to. Or you can just buy plates from a vendor that is already pre-coated. Now this is the most expensive option. So what you do is you try A, then you go to B, and B doesn't work, then you go to C. Does it really matter which one you use? You use what's best for whatever experiment you're trying to do. In some cases, it is just fine to find the minimal, most inexpensive method, because you just want your cells to survive and divide, and whether they're bound to what they're maybe bound to in vivo is not as important to what you want to do. But in some cases, it could be like, well, I want to do option C and I want to create an environment that closely mimics the in vivo environment because I'm testing something which this is important to it. So then you would have to maybe coat the bottom of the plate with different types of proteins to create something that's closer to the in vivo environment. Not adherent cells. Cells that are not epithelial origin, such as cell cultures of hemopoietic origin, lymphocytes, and transformed cells of epithelial origin that no longer require attachment to surface to survive and grow. So these two are sort of self-explanatory. These are cells that sort of go through your lymph and the blood, the circulatory system. That's where they live. That's where they survive. They don't need to attach to survive. They will survive just fine on their own. The last one here is basically if you have cells that are epithelial origin, but they have been transformed and they're at a level of malignancy, which you typically see with cancer cells that metastasize. In that case, some of these cells have basically gain the ability to always have those trophic signals being sent into the cell in the absence of binding to a basal membrane, to a basement membrane. So all that means is this. Let me actually draw this out. Normally, our cell needs to bind to a basement membrane, epithelial cell, and to get these trophic signals coming in, right? That tell it, hey, don't commit apoptosis. Well, a very advanced cancer cell of epithelial origin will be able to not be bound, but yet still receive these signals. Right? This is through mutations and etc. So in this case, these cells can enter the circulatory system and then metastasize because they can seed themselves in a different part of the body. So if you have cells that are of that type, then you don't have to worry about having them bind. They might not even be able to bind. Maybe they're just happy not binding. So it just depends. The interesting thing, I guess, the side thing to say is that cells, basically the apoptosis machinery is ready to go at all times. Signals are coming into the cell, keeping apoptosis at bay. So apoptosis is sort of like a car that is on a downhill, facing a downhill, and one foot is already on the accelerator, and the other feet are these trophic signals coming in, basically keeping their foot on the brake so the car doesn't move forward. So every time trophic signals are lost one by one, the pressure on the brake keeps decreasing until the acceleration takes over and the thing just goes crazy and apoptosis is triggered, and it's a very quick event and the cell dies. Non-adherent cells do not require treated surfaces and can be grown in plates or flasks with hydrophobic surfaces. Well, you don't need to bind to anything, so you don't need to worry about the bottom of the plate. Okay, we'll stop here and continue later. Thank you, guys.